Good morning, good afternoon, good evening, good night, wherever you are in the world. My name is Simon Timperley from the International Food Safety and Quality Network, and our special guest today is Wilfredo Ocasio from Eurofins. Hello, Wilfredo. First time with us. Nice to have you. How are you doing?
I'm doing great and very pleased to be here today, Simon.
Yeah, and you're actually morning. You're up bright and early. Tell the audience where you're joining us from today, Wilfredo.
I'm in Livermore, California, which is the San Francisco Bay Area. So it is early in the morning. I'm still in my first cup of coffee today. So yes, it is perfect.
And I'm, if you don't know, I'm in Manchester in the UK. It's 3 p.m. And it's good to see you in the sidebar. Good evening from Saudi Arabia, Nada, from Minnesota, Dallas. Yeah, wherever you're from, let us know in the sidebar, but you're all very welcome. Food Safety Fridays, we provide you these short bursts of education. You get a free certificate of attendance, and they are sponsored. So I'm going to play the sponsor ads now, and we'll be back in 2 minutes for the presentation. Okay.
The world of food has changed a lot in the last 100 years. But one thing that doesn't change? Ensuring the quality and safe handling of food. No matter what changes are yet to come, we're proud to always be on our clients' side, shaping the future of food today and tomorrow. AIB International, ever onward.
Okay, we're back. Thanks to the sponsors. Just to say, it is being recorded today, and we'll follow up with an email afterwards with the recording slides and your certificate of attendance. It's really nice. We've got almost 400 people live with us now, 2,000 registered. Really looking forward to this topic. So I'll be back later for the Q&A, but for now, I'll hand you over to Wilfredo.
Hey, thank you, Simon. So the topic today is validation of microbiological control measures, and what we're really talking about is the kill step, pasteurization, sterilization step that is applied to foods to eliminate unwanted microbial pathogens and in some instances spoilage organisms, but the focus today is definitely on microbial pathogens. In terms of agenda, I just want to say a couple of words about my company, Eurofins, to begin with, and then go on to discussing the difference between validation and verification, which are two terms that are often confused and misunderstood. Then we'll move into the why, when, and who is responsible for the validation. What do we really validate? This is where it's really, really important to understand the product and technology so that you can adjust the validation accordingly. And then we'll go into the how piece of validation, and particularly using a case study focusing on the thermal processing of low moisture foods, or LMF, which have been a product category that has seen a number of outbreaks and recalls particularly in the US in the last few years. So keep moving. First, about our company, we were founded in France in 1987. We offer testing services, training and consulting, auditing and certification. We also have a very large and strong product design, product development team. Obviously, the R&D function with a force of 40,000 employees in 47 countries and 800 sites around the world. The group that I belong to is a group of microbiologists and engineers, and we focus on helping food companies assess microbiological safety and regulatory compliance for their products and or related processes by conducting laboratory-based research studies and on-site process validation. So we offer a variety of microbiological research studies, including challenge studies, thermal death times. We're able to work with Clostridium botulinum, etc. But let's move into the core of today's presentation, which is validation of microbial control measures. And I wanted to spend a good amount of time making the distinction between the term validation and the functions of validation and that of the verification piece. In fact, these two terms have been often confused, and particularly in the United States, following the promulgation of the Food Safety Modernization Act, which was a new law that came into effect in 2011 in the United States. There was a lot of discussion as to what the requirements pertaining to validation would be. And to that effect, we sponsor, along with the Institute of Food Safety and Health, a gathering of 150 industry and academia experts to talk about validation and verification and the different functions, particularly as it pertains to the preventive control rule under the Food Safety Modernization Act, or FSMA. Those proceedings were published in scientific journal, Food Protection Trends, in 2014. And I just wanted to put this up here, along with a reference to a publication, to make the point of how important and how prevalent was this confusion and interest in understanding the difference between the two terms. So let's go over the definitions as presented in the Food Safety Modernization Act, the Preventive Control Rule. What is validation? Validation, as defined in the aforementioned document, is obtaining and evaluating scientific and technical evidence that a control measure, combination of control measures, or the food safety plan as a whole, when properly implemented, is capable of effectively controlling the identified hazards. While verification is the application of methods, procedures, tests, and other evaluations, in addition to monitoring, to determine whether a control measure or combination of control measures is or has been operating as intended, and to establish the validity of the food safety plan. Now, that is a very big mouthful, and what I like to do is try to digest down to more clear or layman terms. So validation, in my view, are the control measures, or is the function to answer the question, are the control measures the right ones? Will they work? Is mostly associated with the initial installation or implementation of a new control measure. It looks at the efficacy of that control measure, and it may involve some research to understand whether the validation approach is adequate for the intended purpose. Monitoring, on the other hand, tries to answer the question, are you staying within the critical limits that you have established as part of your HACCP plan? This is more in real time, it's continuous, it's during production. While verification is about confirming that you say what you do and that you do what you say. And it's more of a periodical function to make sure that the control measures are working. When I think of validation, I think of validation as being essential to assure public health. It is a regulatory requirement in the US and in many other countries. It has to be done right by the right team. And I'm going to emphasize the team aspect of this quite a bit during this presentation because it's a team effort. It has to be planned with a proper allotment of time and cost and align with the commercial viability of the product and process. Of course, if it is not financially feasible, it's not commercially feasible to do, then it's just not going to happen. Validation is not easy to plan or execute, and we'll go through a lot of details here today. It's not research and development from the standpoint that you go into the validation phase with the idea of conducting a research project as to whether different concentrations of a given sterilant or different temperature time combinations will give you a given result. You have to go in with a pretty good idea that the control measure will be effective in order for validation to take place in a proper way. And it is not inexpensive, and it's not guaranteed to prove that you're going to obtain the required kill, for example, a 5-log reduction of a given or pertinent pathogen. So let's go into the why, when, and who is responsible. Why do we validate? Well, we validate to demonstrate and document, which is very important, the documentation record-keeping piece of this, to demonstrate and document that the control measures in place are sufficient to provide a microbiological safe and stable product. And like I said before, it also is a regulatory requirement under FSMA and other regulations. So who is responsible for the validation? Under the Food Safety Modernization Act, PC rule, there's a designation of a qualified individual who must do or oversee the validation of preventive controls. This qualified individual must complete training equivalent to a standardized curriculum recognized by the Food and Drug Administration in the US, or be otherwise qualified through job experience. If we look at the International Commission for Microbiology Specifications in Foods, ICMSF, they refer to a team of experts as a requirement for system validation because of the many skills required, such as engineering, microbiology, physical chemistry, etc. And I'm in very strong agreement with this. There's so many disciplines that are involved in the validation process that you do need a team. As part of the publication from the Food Safety Summit that I cited before, I also quoted the phrase or sentence, the establishment of food safety functions, including validation, is a team effort that include corporate managers who set policy and standards, and supervisors and line workers who implement the programs. So the participation of management is essential in this function, as well as the technical and folks working right in the front line dealing with production day to day. When to validate? Well, validation is normally associated, like I mentioned before, with the initial implementation of a control measure. But when changes occur, in that control measure or other aspects, revalidation may be necessary. And I listed here some potential triggers for revalidation. For example, emergence of a new pathogen or a higher risk in the given product, or monitoring or verification shows system failure. A change in ingredient microbial load. You all of a sudden move into a different ingredient that may have a larger propensity to contain salmonella, for example. A change in the product formulation that may result in a higher pH, for example, that would make your thermal treatment less effective. A change in processing parameters or storage conditions. A product category is linked to an outbreak. So you have an outbreak in almonds or in peaches or in onions that may trigger some reevaluation of your validation strategy. A regulatory agency alerts, new information in the scientific literature, consumer complaints. These are all potential triggers for having to revalidate the control measure. It is really important that as you move through production and you move through validation, verification, and monitoring, you have a very strong management of change program, keeping track of all the changes, how your process is evolving, and so on, as this may provide information relative to some of these triggers that may require revalidation. So let's move on to the what is it that we truly validate when we're talking about validation of control measures. And this is where a very thorough and deep understanding of the product and technology is very, very important. Two things to keep in mind as you are developing your validation strategy is that validation is very specific to the intended product, process, and process parameters being evaluated. And validation must be conducted using a worst case scenario in terms of critical process and product related factors. So if you're looking into a product with a given pH or a pH range, normally you would be, during the validation process, you would like to adjust that product to the higher pH, which represents the most conservative scenario. If you're using a range of temperatures on the pasteurization step during the validation process, of course, you want to use the lowest possible temperature that you'd be using during production to create that worst case scenario during the validation. So when we're talking about validation, what you're really validating, obviously, is the control measures. But within the control measures, you're validating the process parameters. Sometimes we call them critical factors, critical control points. You're validating, for example, a given temperature in terms of the ability of that temperature to, and time combination, to deliver a certain microbial reduction. You may be validating a concentration of a given chemical to provide the sterilization of packaging material. For example, if you're using hydrogen peroxide to sterilize PET bottles, what you're validating is that concentration, that time, that temperature provide the required microbial reduction. For example, a 5-log reduction of the pertinent pathogen. And of course, that is the critical limits. You need to conduct the validation at the limit of that you have established as your minimum for delivering the proper result. In terms of understanding the technology, we do a lot of high pressure processing in our research facility. And when you think about high pressure as an example of a technology, and this goes into understanding the technology, is very simple to think that temperature and pressure are the only things involved. But when you go in depth as to what are all the critical factors that you need to take into consideration, I have listed quite a few here that involve both the process as well as the product, and you have to have a thorough understanding on the product side of, for example, of the pH and the water activity of the product. High pressure processing is very ineffective at lower water activity levels. It's also relatively ineffective when you go up to higher pHs. On the process side, you have to pay attention to the starting temperature of the product and pressurization fluid. Both of these influence the degree of kill. You need to look at the time to reach the target pressure, or that ramp up to get to pressure. But the point here is not for me to list all of this, but is to convey the message of the importance of understanding the technology and the product. And these are just some examples of actual results from high pressure research that we've done in our laboratories related to the destruction of E. coli, Salmonella, and Listeria monocytogenes. High pressure processing is widely accepted for processing of beverages and juices, and yet we saw here that with different processes, and when you look at these graphs, the dotted line represents a 5-log reduction of destruction of these organisms, and when you see the bar going above the dotted line, it represents that you have achieved the 5-log reduction. But as you can see, in many products, and particularly in low-acid beverages like carrot juice, we had a really hard time, even at 85,000 psi for 3 minutes, to get anywhere near close a 5-log reduction of E. coli 157 and Salmonella in certain products. So this is just an illustration of the importance of conducting a validation step to make sure that your control measures are effective. Other technologies that we've used on beverages in our laboratories include pulse electric fields. And again, this is just a quick illustration of how important it is to understand all the different critical factors that play into the microbial destruction in these individual technologies. So this is an example of pulse electric fields. Again, a number of product critical factors. Here are things in the product like ionic strength of the product plays a big role, as well as a number of process critical factors. Other examples that we work with is supercritical CO2, UV radiation, and a number of different others. But again, it's the importance of the product, understanding the product and the process. So moving on, let's talk a little bit about the validation approach. How do you validate? How to validate? In my view, and using guidelines from the United States Department of Agriculture, Food Safety and Inspection Services, there are two important elements to validation. The first one is to gather scientific or technical support for the control measure. This is the understanding that what you intend to do has been proved scientifically to be effective. For example, you are estimated that a given temperature and time combination is effective against Salmonella in say banana puree or in another commodity. Well, are there information in the scientific literature? Have you done challenge study or thermal destruction studies that demonstrate that that is an effective way of doing the work? Then the second part of the validation is to demonstrate that in the plant setting, that control measure can deliver as expected, or perform as expected. And both of these elements must be completed. In some cases, element 2 may serve both purposes. You may develop the scientific and technical support, as well as do implant, as well as doing the implant demonstration. And it's important to not to confuse element 2 with verification. It is also important to understand that validation may be accomplished in different ways, and particularly in two main categories. One is collecting and evaluating scientific and technical information. This may come from historical knowledge. This may come from published safe harbor processes. These are processes that apply to broad categories of products. This may be based on regulatory or performance standards or published processing guidelines. It may come from the peer-reviewed scientific literature or technical data, from previous studies that have been conducted, or from scientifically developed mathematical models. One thing to keep in mind is that in all these cases, the data must match the specific product or technology being validated. So if you have data, for example, to a formulated beverage that contains as an acidifying agent citric acid, and you're trying to apply that to a formulated beverage that contains a different portfolio of acids, the data that is in the literature may not truly match that of the product that you're applying it to. So you have to be very careful with that. And it's something that here in the United States, the FDA pays a strong attention to. The other way of conducting validation, and the way that we are emphasizing in today's presentation, is conducting validation studies. And this is collecting scientifically valid experimental data on site, or conducting challenge studies or thermal death time studies or inoculated packs in the lab. You can also use some of the elements of monitoring and verification during a defined validation period in the establishment to serve as a validation process. Okay. And like I mentioned at the beginning, let's use low moisture foods thermal process as an example or case study for a validation effort. And before we go in depth, I want to run through some definitions in terms that I'm going to use in the next few slides and that are important when you're validating thermal processes. The target organism is a pathogenic organism, Salmonella, E. coli, Listeria monocytogenes, that is significant in the given product. This could be Clostridium botulinum in the case of shelf-stable low-acid products that are canned, for example. The surrogate organism. The surrogate organism is a non-pathogenic organism which mimics the resistance of the target organism to the given technology and is appropriate for use in validation work. So these are organisms that you could take into the production facility, run through the control measure, and demonstrate the destruction of the technology on site without having to take a pathogen to the production facility, which obviously is not a viable option. Process lethality is the measure of the process ability to destroy the target organism, and this is normally expressed as a log reduction. So here in the US, the regulatory agencies, when it comes to vegetative pathogens like Listeria, Salmonella, E. coli, they really focus on this idea of a 5 log reduction. If you're talking about shelf-stable, low-acid products like in a can environment, Clostridium botulinum, the term 12 log reductions, or the target of 12 log reduction, is normally in place. The kill ratio is something that we use to correlate, it's a term that we use to correlate the destruction of the surrogate organism to the destruction of the target organism. So it's, in a more formal definition, the mathematical correlation between the destruction of the surrogate and the target organism. And this is where understanding of the D value and the Z value, which are the rate or kinetics of microbial destruction, is important when we're talking about thermal validation. So you have D value is the time in minutes at a constant temperature necessary to destroy 90% of the microorganisms, while the Z value is the change in temperature necessary to bring out a tenfold or 1 log change in the D value. So since we're talking about low moisture foods, I would like to make it clear what we're talking about. Low moisture foods are foods with a water activity of 0.85 or below. It's important to understand that even though I'm using the term low moisture foods, this term is being used interchangeably with the term low water activity foods. But in reality, we're talking about low water activity foods from a purely technical standpoint, because moisture, which is the amount of water present in the product, is not the same as water activity. So water activity is the ratio of the vapor pressure of water in the food matrix compared to that of pure water. Water activity is a measure of the amount of water that is chemically bound and hence biologically unavailable. So at water activities below 0.6, for example, there's no microbial growth. At water activities below 0.91, most pathogenic bacteria are inhibited, with the exception of 0.85. So most low moisture foods have, when I'm referring to low moisture foods, I'm referring to foods that have a water activity between 0.4 and 0.85. Example of low moisture foods go all the way from dry fruits, cereals, confections, peanut butters, dry protein powders to egg powders, etc., syrups. And the processes vary from drying, like for example, dry pasta, to extrusion, as you would on dry pet food, to dry roasting of almonds, to baking of cookies, pastries, other drying processes, processes prior to drying. Some of these products are pasteurized on the wet side, like milk for drying, for example. One thing to keep in mind, and one of the big challenges that we find when we're doing all of these validations, particularly with this legacy equipment like drying equipment in the dry pasta industry, is that most of this legacy equipment was designed to deliver a certain product functional characteristic, not necessarily as a pasteurization or kill step. Therefore, it may not have all the sensors and ability to monitor the pasteurization or sterilization as you would in equipment that was designed for that purpose, like for example, an UHT treatment in the aseptic processing side, or pasteurization treatment in milk. Validation is essential to public health in low moisture foods. Low moisture foods were normally in the past considered to be a low risk product, because at these water activities, most of the pathogens will not grow in the product. But unfortunately, they may survive for very long periods, 12 months in almond kernels, for example, at a water activity of 0.4, 24 degrees C. These pathogens that can survive for long periods, for example, salmonella, they do have a very low infection dose. So even though they do not grow in the product, they may still cause illness at very low concentrations. To make things even more complicated, these pathogens demonstrate much higher thermal resistance in the low water activity environment as compared to the high water activities environments, as you would find in products such as meats or milk, etc. And when we're talking about low moisture foods, salmonella is the most prevalent issue, albeit E. coli 157 and others may cause problems as well. I have numerous outbreaks that we can cite here in the United States, and I'm gonna go through those, but that implicated low moisture foods, as you can see, mostly salmonella and E. coli 157A7. So when we get into the actual how of the low moisture food process or control measure validation, we like to divide this into three major phases. One is the product process assessment, building the foundation of how you're going to go about the validation. Then as a phase 2, we normally like to, particularly if we're going to use a surrogate, do some microbial kinetics in the lab, developing those tools, understanding the resistance of the pathogen, understanding the resistance of the surrogate, so that we can create that correlation between one and the other before we go into the food plant and execute the validation. And then the final phase is the process validation, executing the validation study at the facility with the final result determining the log reduction that the control measure would deliver. So in terms of phase 1, understanding the product and process and what we refer as building the foundation, have to understand the regulatory requirements. You have to define individual responsibilities, who's going to be part of that team that is going to be running the validation. What are the target organisms? What are we going after here? Salmonella, E. coli 157. Are we going to be using some surrogates like Enterococcus faecium, which is being commonly used as a surrogate for Salmonella in low moisture applications? Understanding the process, like I've emphasized in this presentation so much, understanding the equipment being validated, the manner in which the heat is applied, the temperature and time, and not just only the setting on the equipment, but it is really important, if at all possible, to understand the product temperature profile during the actual process. The order of introduction of ingredients, this is really important. We've done a number of extrusion processes, and ingredients are added at different points during this extrusion process. So understanding what is the most conservative approach, what goes into the extrusion process last and receives the least amount of process, for example. If you're using a conveying belt, the depth and distribution of the product and that conveying belt that goes through the pasteurization tunnel, for example. There's a number of other extrinsic parameters, the relative humidity, the airflow in a drying operation may be very important. And understanding the product, like we talk about moisture, water activity, fat and oil content, etc. The presence of antimicrobials in terms of physical properties, piece size, shape, structure, surface area are all important. And all of these feed into identifying the critical control points and critical limits that you need to assign to each one of those values. In a drying environment, for example, we have the challenge of the product changing water activities as it moves through the different zones in the drying. So when you're establishing the resistance of a given target organism in the given product, you have to make decisions as to what water activity you use in those studies. In our lab, we tend to use the most conservative, which means the lowest water activity, to determine the resistance. But it is important to understand that this changes as a function of the process. So here's some examples of critical control points and limits, which we already covered in the previous slide. But all of these have to be established before the validation itself begins. Understanding the temperature profile in the oven or drying is really important. It may be something that can be done prior to starting any microbial validation, and in some cases in lieu of the microbial validation using a surrogate. If you can establish, for example, temperature data on the product itself, and there's different ways to do that. In some cases, it's possible to do. So all of that goes into the preparation to the validation. Here's a graph that shows the relative resistance of a surrogate versus the target organism. Like you see here, the surrogate is more resistant in terms of temperature, so it's an appropriate surrogate. Then we go into phase 2, which is understanding the resistance of the microorganisms in the lab. Okay, and like I mentioned before, Enterococcus faecium and Pediococcus have been widely used. This work have to be very specific to the product and the organism that obviously that are being evaluated. And the results of these studies is a DNC value that allows us to compare the surrogate to the target organism. And surrogate organisms have very specific characteristics that are important. First of all, they must be non-pathogenic. Ideally, they're stable enough so that they can be transported effectively from the laboratory to the test site. They should be easy to grow to high levels so that you can conduct an effective validation. Easily differentiated would be a nice thing to do for the lab, identifying the different microorganisms. And obviously, they should show similar and quantifiable resistance as compared to the target pathogen, okay, to allow for comparison between the heat resistance of the surrogate and the target pathogen. So it is really important when you are doing this work to do it appropriately and a lab that is qualified to do this type of thing. We in our lab follow the National Advisory Committee for the Microbiological Criteria for Foods guidelines to do these studies. The growth conditions that you use to prepare the organism that you're going to use are really important. If you, for example, are working on an acid environment, you must acid adapt to your microorganisms before you use them. For example, the inoculation method, if you're working with low moisture foods, you must inoculate in a way that doesn't impact the water activity of the product. The heat treatment method that you use is important, and the recovery that you use after the treatment to determine the survival of microorganisms after the treatment is really important. For example, avoiding using selective media, which may impact the survival of injured microorganisms after the process and give you a false negative result. So these are some examples of the type of data that comes out of this type of studies, where you can see the effect of time at a given temperature on the survival of the microorganism. These allow us to plot the resistance of the surrogate versus the resistance of the target. And this is one example where you see that on the top portion of this range of temperatures, we see that the surrogate is much more resistant than Salmonella. But as we move up in temperature, because of the differences in Z values or the slope of these curves, the reverse is true as you move to different temperatures. Then the final step, phase 3 in the process validation, is to introduce the surrogate organism into the actual product, and doing so without changing the intrinsic properties of what you're doing, having enough inoculum to be able to quantify it through the process. Sometimes this involves using coloring, food coloring dyes to identify the portions of products that are inoculated, for example. Some people use sachets mesh sachets to capture the inoculated product. This is generally not recommended, but is used. And the inoculant should be stable. The entry point, like I mentioned before, in the process have to be conservative, and you have to use worst case processing conditions, and make sure that the inoculant portion, the inoculated portion of product is placed in the cold spot of the process. So if you have a temperature profile throughout the pasteurization process that reflects a cold spot or a lower temperature area, that's where you need to have your inoculated portion. Then the final step is to do your lethality calculation. You have information as to the load of surrogate Going into the process, you determine the load of surrogate coming out of the process, and you understand the correlation between the surrogate and the target organism. So once you have all of this information, you can make a determination as to what the log reduction was for the given target organism. And that was my final slide. I thank you very much for your attention. And now we'll move into the question portion of this presentation.
Perfect. Thanks very much, Wilfredo. If you switch your webcam back on, there's been a heck of a lot of questions. I don't know if you noticed, but I'll just pick through some and see where we get to. Right. So, Mohammed, one of the validation approaches to do research or consider research literature as per Codex validation guideline, is it not? Does that make sense to you?
Yes, absolutely. Codex is a great resource. There's a number of guidelines on the U.S. Food and Drug Administration related to compliance with preventive control rule. There's a National Advisory Committee for the Microbiological Criteria Foods. They have an excellent publication on validation of different control measures, pasteurization measures, and so on. So you can go to any of the aforementioned website and find guidance in that front.
Okay. Next, Chandrasekhar. If revalidation is not done as per the potential triggers shown in slide 13, can we raise as a major non-conformance during the audit?
Absolutely. So if there's a trigger that calls for revalidation, for example, you're changing some intrinsic parameter of the product. I mean, is you've done validation that demonstrate that the process is effective at pH 4.2, and now your product replace a product that contains a pH of 4.4. Now your pH has drifted up, and you have not revalidated that control measure. Absolutely, you could have an uncompliant situation. So, and there could be like I had on the slide, a number of different triggers that would cause that situation.
Okay, thank you, Rosanna. If nothing changes in the operation / control measure, for how long could a validation be valid to be considered? Does it have to be performed every start of a new season? Is there a frequency?
No, absolutely not. What validation is something that you do initially to demonstrate that the critical limits that you put forth are effective in providing the given result. So once you get that established, what you would do is making sure that that is in place all the time by monitoring and verification functions. So the Food Safety Modernization Preventive Control Rule, for example, in the US requires re-evaluation of the food safety plan every 3 years. But this is different from validation. It's an evaluation of the plan to make sure that there haven't been changes, that everything is under control, etc. Now, as a result of that re-evaluation, you may discover changes that one of the triggers have been activated, and that revalidation is required, but is not something that you would do periodically like you do verification, or do continuously as you do monitoring.
Okay, there was a couple of questions, but Bruce answered those kindly, so we'll skip past those. Asif, I have a question about Eurofin's laboratory. Does your company have ISO 17025 certification?
Yes, absolutely. But I should clarify that in the research function, for example, research doesn't fall under ISO 17025. Is more under ISO 17025, you are certifying methods, procedures, SOPs for salmonella testing, for example. So we're in an environment where ISO 17025 certification is there. we may not be applying 17025 for a research project, correct? But everything that goes into the research project, the media, the incubation, everything is done in an ISO 17025 certification environment.
Okay, thank you. Sarah, how can you control listeria in high moisture products, i.e. runner beans?
Well, there's obviously there's a number of different control measures in this environment. What we call prerequisites, good manufacturing practices are really important. The sanitation process, even though under the regulations, sanitation processes in the US do not necessarily need to be validated. Most industry folks go through a validation of the sanitation processes. And of course, the implementation of a good environmental monitoring program is I believe it's an essential in control of listeria in this high moisture environment. So is in particular, if you don't have a kill step, or if you have a kill step followed by potential post-process contamination, you have to have a group of control measures acting together to alleviate this issue related to listeria.
Great, thank you, Wilfredo. Emily, quite a long question. I'll read it out for those that see this recording after without the sidebar. If a facility places spices, dried vegetable matter, etc. into buckets and fills the buckets with hot water as a treatment for the inclusions, would you please explain what would need to be done to validate the process? And can you give us an example of what a proper validation would consist of? What would be the verification monitoring activities for this process?
Okay. Well, the first thing that I would recommend is that you have to have an understanding of the temperature profile of the different inclusions in this bucket. Obviously, it has to be consistent, it has to be controllable. But gathering temperature data, and particularly if you have pieces that stack together or pieces that could be contaminated more in the center, this may have to be impelled by the temperature sensor to gather interior temperature. And once you have the temperature and time profile for the different products that go in there, then you can look at where you're at relative to the resistance of microorganisms. You could be in a very conservative space relative to destroying Listeria, Salmonella, E. coli, meaning temperatures in the 90 degrees C or something along those lines. Or you could be very marginal, where you have much lower temperatures in certain cold spots, and you may have to do some work in the lab to determine whether those temperatures would be effective against that particular pathogen in the given product. So temperature profiling would be the first thing that I would recommend to gain. Understanding the process is a lot of what we talk in the presentation, and that's what you would need to do first.
Okay, thank you. And Laurie, validation challenge studies specifically is typically best completed by an unbiased accredited lab. Can you make some comments around this?
This is absolutely important. I mean, we see in the US, we have what we call under FDA filing something called formulation control process. This is where you preclude the thermal process in some instances. Say, for example, an acidified salsa product, where you have antimicrobials and pH, etc., controlling the survival of pathogens in there. And in fact, when you introduce pathogens, they die off rather quickly. So how you demonstrate that is the case is by conducting a challenge study. These have to be done properly. Like I said before, you have to adapt the microorganisms to the acid environment before you introduce them. You have to recover them in non-selective media to make sure that you're not creating a bias on the injured microorganisms by putting them directly into media that contain antibiotics or other components that may enhance the process and therefore give you false negatives. So absolutely, you need to have somebody that knows what they're doing.
Okay, and Penny, what do you think of salmonella testing on powder ready-to-drink products? Did decision tree and inconclusive. No kinda off topic, but company specific. Any comments on that one, Alfredo?
I'm trying to understand the question.
Yeah.
Finished product testing could be part of your food safety plan, particularly when there's not a lot of, there's not a kill step, but it cannot be the only thing that you relied on. You have to have, an ingredient supplier verification program. You have to have your environmental monitoring. You have to have your sanitation, etc. And then collectively, as part of your food safety plan, you have your finished product testing, and that collectively will provide you the protection. But testing alone will not be enough, albeit it could be a very important piece of it.
Okay, and Laurie, please comment on micro modeling that is available that could be investigated and used if appropriate.
Absolutely, yeah. So CONT-based and other scientifically based mathematical models are available and can be used either to validate the process itself, if you can create a reasonable and defensible scientifically correlation between what was used in the model and your product. They're a good match. But most of the time we use them as a guidance to adjust the critical limits to values that have a good chance of being successful, and then we go into the validation to prove that they are indeed successful within the context of your product and technology. Okay. But they're very useful tools, yes.
Thanks. And Sharon, on slide 37, if the surrogate is behaving differently than salmonella, how can it be a good substitute for salmonella and give reliable results?
Okay. So, the classical example of use of surrogates are in the thermal processes, right? And what we're like the example in the presentation. So what you do is that you establish the resistance of the surrogate to the given process in the given product. So you have to, it's specific for the technology and the product. And then you establish the resistance of the surrogate. So you have this correlation, and you may understand that that they're correlated 1 to 2, for example, meaning that if you destroyed 1 log of the surrogate, you'll be destroying 2 logs of salmonella. So if your target is to destroy 5 logs of salmonella, obviously you look for a 2.5 log reduction of your surrogate during your validation process to demonstrate your target. So, but you have to have this understanding how the surrogate and the target organism behave in the given technology and the given process, and establishing that correlation, which we call the kill ratio between one and the other.
Okay, great, thanks. Vicky, another quite long one. When there are outbreaks with a new or rare strain, are surrogates re-evaluated against those? I'm wondering about the recent salmonella outbreak in onions. Any concern with current validated processes for dried, dehydrated onions?
Well, this definitely falls in that category of new information that comes into play in the literature or via regulatory or outbreaks, and you do have to throw that into the equation, particularly if you are doing validation within the commodity that was affected, for example. So absolutely, it would be a good practice to go back and soon enough there will be information in the literature with the strains that were implicated if they're not in strains that we're familiar with, and if by some odd reason, a good example was years ago when E. coli 157A7 came into the food safety environment. We did not have a good understanding of the acid resistance of this organism, and that's changed a lot of what we did challenge study wise in acid products, for example. So yes, it is very important to keep that in mind as you go through your process. And I'll say hello to Vicky.
And Camilla, in a validation process, do we necessarily need to use surrogate microorganisms, or can we just rely on product regular lab tests?
No. So product regular lab tests will not replace validation or count as validation. So, however, you can do validation in some instances by collecting samples during a given processing time at, when you first introduce a control measure. So using the tools of validation and monitoring may be a way to go about doing the validation. Is something that is a little bit tricky, but definitely you cannot rely just on product regular lab tests as a substitute for validation. No.
Okay, and E. faecium is less heat resistant than Salmonella at temperatures higher than 78 Fahrenheit. Does that mean we have to choose another surrogate for that temperature range?
Well, I cannot tell you. It depends on the product, whether Enterococcus faecium is always more or less resistant than Salmonella at a given temperature. But yes, we do this all the time. We do, before we go into the field with a surrogate, we do what we call a little, the comparison between the two. And when it doesn't work, one thing that you can do, meaning that it is less resistant, the surrogate is less resistant than Salmonella, sometimes you can compensate by using higher inoculant levels of the surrogate. Let's say that the correlation is reversed. So one log destruction of the surrogate only translate to half a log of Salmonella. Well, then you will have to destroy 10 logs of the surrogate. And if you can get to a condition inoculant-wise where you can get to 10 log, which is unlikely, but you can do that calculation, you could use the surrogate at a higher concentration. But what we do most of the time when that situation arrives is, yes, indeed, try other surrogates.
Okay, and Rancy, is it worth to do any validation if no pathogen risk is present at the starting point only?
I'm not sure what is meant by the starting point only, but obviously the one thing that I didn't talk about because there was not enough time, but the hazard analysis as part of your HACCP or hazard analysis critical control program is a very important piece that is very connected to validation. During the hazard analysis portion of your food safety plan or your HACCP program is when you're going to determine what are your microbial risks. And once you identify that, then you assign control measures that address those specific hazards that you have identified. So you have to conduct your hazard analysis, and if you're claiming that you don't have a pathogen risk, you have to justify it scientifically. I don't have to worry about E. coli in this product because I receive it from a supplier that these are aseptically processed, they're thermally processed, they come to me as commercially sterile product, and I know what they're doing. I know that they're I have a supplier verification program that tells me that they're doing the right thing, so I don't have to count this as a hazard on my operation.
Okay, and Annabella, one of the slides mentioned Salmonella was higher resistance at low WA. Can you explain why is that?
Okay, so it's not just Salmonella. Almost every microorganism is more resistant as you lower the water activity of a product. So dry heat resistance is much higher than wet heat resistance. And the classical example for that is that in your kitchen, your oven is at 400 degrees Fahrenheit. You can stick your hand in the oven without touching any surface and you don't get burned. But if you put your hand over the boiling water and you get hit by the steam, you get a much greater burn. That's the difference between dry heat and wet heat. Wet heat, much more effective in terms of the destruction of microorganisms because of the heat transfer coefficients involved are much different than in the dry heat environment. So that's a common thing, well-known physical phenomenon, and that's what I was talking about.
Great, thanks, Alfredo. Anna, what is the recommended target microbial load reduction when validating a cleaning method applied to say that it is effective?
Well, when validating a cleaning method, that is different. There, what you're looking for is removing organic matter, removing debris. So you can evaluate that by different methods that do not involve microbial load, like ATP measurements and things of that nature. If you're looking at an intervention or control measure in terms of microbial load reduction, like I said in the presentation, to be effective, the 5-log reduction of vegetative pathogen is a commonly, widely, globally accepted number. But for cleaning, it's a different thing. It's not the same as sterilization or pasteurization. So if you're referring to cleaning, then you use cleaning assessment tools, like I mentioned.
Okay, Helena, in the case of an organization that does catering, how would validation of the microbial control measures look like in order for it to be adequate, especially when venues of serving change, i.e. customers?
So this is if we're talking about food service or catering, that's a different environment than the control measures that we were evaluating, but in a food production facility. But if you're transporting food, for example, and that type of thing, obviously temperature control and your ability to control the temperature. If you identify temperature control as a control measure that is critical for the safety of microorganisms, then you better have the monitoring and verification capabilities to demonstrate that you are able to maintain that temperature throughout the distribution effort or during the presentation and catering function. So.
Okay, and Rose, food regulations require pathogens like Salmonella to be negative in the finished food products. What is the appropriate validation for this?
Well, again, the 5-log reduction. So you want to demonstrate that whatever intervention you have is, if you're going to call it a kill step for Salmonella, you have to demonstrate that that intervention or control measure translates to a 5-log reduction.
Okay, and Arpan, what should be specific requirements for validation for foreign suppliers who are exporting to US and may not follow FSMA?
You have to follow FSMA to export to the US. So it's not one or the other. I mean, you have to, you have to. I don't know how else to answer that question. You have to follow FSMA. You're bringing product to the US.
Okay, Julius, do you recommend using an outside firm like Eurofins to perform a validation study? Sure you do you?
Yes, yes, I do.
Of course.
Yes.
Marvi, what is the surrogate normally used for Listeria monocytogenes?
Okay, so for Listeria monocytogenes, it depends. It depends on the food product. Like and I had a slide that show a number of different surrogates for different applications. But whatever the surrogate may be, and it could be Enterococcus faecium, it could be Pediococcus, it could be a different lactic acid producing bacteria. You just have, for the given product and technology, you have to establish the correlation between one and the other, and you do that in the lab prior to going to the operation.
Okay, we're going to carry on, if you're all right, Wilfredo, till 1:15, but we can't continue because I'm still halfway down the scroll. It's not changing, so we'll just keep going till 1:15.
No problem.
Okay, VC, Any good guidelines on Clostridium botulinum challenge studies in bakery products with modified atmosphere packaging?
Okay. So the National Advisory Committee for Microbiological Specifications for Foods have a guideline for Clostridium botulinum studies. You just follow those. I mean, there's the type of strains that you will use. Mostly this would be what we call proteolytic strains. You have to have an understanding of the package that you're going to be using and make sure that you're in a conservative environment. But again, what we call the National Advisory Committee guidelines and VC. If you want to reach out to me, I can probably give you the reference. My email is in the presentation. I don't know, Simon.
Yeah, we'll circulate it after.
So so I can give you the citation. But if you Google National Advisory Committee Microbiological Specification for Foods Clostridium botulinum challenge studies, I think you'll get to it.
Great, thank you. Atafe, what are methods to eliminate microbial parameters of food products, pistachios? Are ozone or ionization effective? Ionization in pistachios specifically.
Ozone has been used for many different applications. The efficacy, and I'm not aware of the efficacy or have knowledge about applications of ozone to pistachio and their efficacy. So I'm afraid that I cannot comment on that one.
Grizel, pathogens that should be monitored in a coffee plant. Anything?
Okay, so I assume that we're talking about coffee as a beverage, and here it depends on what process you're applying. A lot of cold brew type coffees now receive very little or no thermal treatment. One of the big questions is whether or not Clostridium botulinum, if this is not refrigerated, what the impact would be. Coffee is not a very good media for the growth of microorganisms, and in fact they die fairly quickly in coffee as long as it's plain coffee. Once you add other ingredients, you add sugar, milk to it, you're in a different environment and you're in the same as any other food in terms of what is your kill step. You have to be worried about your common vegetative pathogens, and in some cases obviously even Clostridium botulinum. Even if it's a refrigerated product, the potential for abuse with Clostridium botulinum plays a role. If you're doing environmental monitoring, Listeria species are a common pathogen or indicator organism to look after. But I would focus on the kill step and evaluating the risk of your product. Like I said, whether it's plain coffee or coffee with additive.
Okay.
And conduct your hazard analysis. Yeah.
Okay, so pathogens don't like coffee so much. Do they like tea?
Tea is very similar with their content of catechins and that are also effective as antimicrobial properties, again, without any additional additives to it that would increase the nutritional value and decrease antimicrobial properties. But coffee and tea are pretty antimicrobial by themselves. Now, I don't want to mislead anybody into thinking that you're completely out of the woods, but that's the scientific information that is out there.
Okay, and Kirsten, if following the recommendations of a process authority, do we still need to validate?
The answer to that, it depends. Okay, so process authorities still have to follow the same rules of validation as you would. For example, if I'm giving you a recommendation for a process in terms of temperature and time, that recommendation has to be backed up somehow scientifically. And I mentioned the number of different ways that this could be. It could be through information in the peer review literature. It could be from information from trade associations. It could be from information historical knowledge. So, for example, in acidified foods, we have a widely accepted process called the F16200. Everybody knows that this is, including the regulatory agencies, that this is a conservative process relative and is well validated over years for these high acid products. So yes, the process authority could give you a letter of recommendation. Just make sure that the process authority himself have the basis to put that forth and he can defend it scientifically.
Okay, thanks very much, Wilfredo. We're going to have to leave it there because we're never going to get through them. It's impossible. But Bruce has made a comment. Hey, Simon, this has been a very good webinar. You will have to invite the speaker back for another go around. So
thank you.
Yeah, it was super presentation. I know you condensed it into about 40 slides. From how many slides, Wilfredo, was there?
I had 105 to begin with, with the different.
So it's really hard to condense quite a complex topic down, and then lots of questions, and they're quite complex questions as well. So you've done a great job, Wilfredo. We will have to have you back next year if you will join us again. In the recording we send the presentation will be there with your contact details. So if you have a question, Wilfredo, he might be able to help. Or if you need some help with from Eurofins, I'm sure they'll help. So, yeah.
Well, I just want to say thank you to the wonderful audience. Thank you for all the questions and for staying throughout the presentation. And thank you to you, Simon, for the invitation. It was a great pleasure.
Great. We'll see you again. We'll give you a shout towards the end of the year for next year.
I'll be looking forward to that. Thank you very much.
Thanks, Wilfredo.
Bye now.
Okay. Wilfredo Ocasio, a real expert there from Eurofins. Delighted to have him along. I'm sure you'll agree. I'm sorry that we couldn't get to all of your questions, but it's just impossible. 2,000 people registered, 500 online, but fantastic. So I loaded your certificate in the sidebar. I'll follow up. Just give us an hour to package it up, the recording, the slides, certificate. In there will be Wilfredo's contact details and Eurofins contact details. So it's Friday, best day of the week. Enjoy the rest of your day. Have a great weekend, and we'll see you actually next week. Watch out for your emails. Thanks a lot. Bye-bye.